Hi, so welcome to this session of 5 Days of Stem Cells. I'm Claudia Miranda. I work at Institute for Bioengineering and Biosciences, and today in our session we are going to talk about how can we develop a robust and scalable culture platform either for expansion and neural induction of human pluripotent stem cells in suspension culture. So human pluripotent stem cells have been thoroughly studied in the past few years. So they are very special because they, for one, they can self-renewal, and on the other side, they can also differentiate into any type of cell of the human body. So we have two main sources of human pluripotent stem cells. The first ones are the embryonic stem cells, which are derived from the inner cell mass of the embryo, and the other ones are the induced pluripotent stem cells, or iPS cells. So these cells are derived from somatic cells that are reprogrammed back to an embryonic state-like. So these cells can be cultured either as pluripotent stem cells in the lab, or they can be differentiated. So, and especially iPS cells have great advantages because they can be used towards a myriad of applications. So since we can derive the cells from a patient with a specific disease, we can use the cells to study the disease in vitro. They also can be used for drug efficacy and safety testing, or even in the future for organ replacement therapy. However, in order to use these cells for any of these applications, we still need to do something, which is we need to obtain large numbers of cells, and also we need to be able to differentiate them into pure populations. So for that, we can use three main culture systems in the lab. So the most classical one is the monolayer, which is basically when we have cells attached to a substrate that is used for cellular attachment. So in this system, we have a uniform application of the stimuli, so all of the cells are sensing the same stimuli at the same time, but we have a small problem, which is achieving high cell yields, because we do have a limited surface area available for cells to grow. So to surpass this, microcarriers were introduced. So these are small particles that can be coated with the same substrates to promote cellular attachment, and that allows us to scale up the production of the cells by increasing the surface area. Still, we need to be concerned about the low adhesion percentages that we may have using these cells, and to use them in another applications, we still need to remove the microcarriers, which will add additional steps on the downstream processing. So, and at the same time, we have more steps and we have increased costs to obtain the cells. So to surpass this, spheroids have emerged in the last few years. So here, we basically have cells attached to each other, which gives the cells 3D conformations. So in here, we are able to better mimic the in vivo environment. So, and as we are not depending on the surface area available, we also can achieve high cell yields at a more cost-efficient manner, since we do not have to add further steps on the downstream processing, or we do not have to buy the substrates or the microcarriers. Still, in the system, our limitation is related with the transport, which arises from size limitations. So in here, when we have aggregates that are bigger than 300 to 400 micrometers in diameter, we may start to observe some necrotic centers as the oxygen and nutrients fail to reach the center of the aggregate, and the metabolites and other gases fail to exit the aggregate. So there's one thing that we need to be concerned about. So, and in this session, the first part of the work is to be able to achieve large numbers of pluripotent stem cells in suspension culture in a robust and scalable way. So for that, we use two platforms. So the first one is the orbital shaker. So we have a culture plate inside on top of the shaker, and the agitation will promote homogeneity in the culture medium and at the same time in the average diameter of the aggregates. So also by using the 6-well culture plates, we are working with smaller working volumes, which allows us to screen a large number of conditions at the same time. But still, we wanted to scale up this up to a bioreactor. So for that, we use the PBS Biotech bioreactors, which has a rotation controlled by magnetic coupling that exists between the vertical wheel impeller, that it's like an hamster wheel impeller, and the base unit. So the shear stress produced by this bioreactor will be lower than the ones produced by, for example, a spinner flask. And another advantage is that we are working with single-use vessels, so it's easier to do the process over and over again. Also, for the two systems, we used StemScale. So StemScale is a newly developed culture media, especially designed for the expansion of human pluripotent stem cells as suspension culture. So we wanted to prove that this medium would be robust enough to allow us to culture the cells under these conditions. So to start, we used the orbital shaker. This shaker, the culture plates in the shaker had working volumes between 2 and 4 mL per well, and we used a seeding density of 150 cells per mL. So to do this testing, what we did is that we seeded the cells as a single cell suspension. We let the cells grow for up to 5 days to ensure that they do not pass the 400 micrometer limit. And we do this for the first passage, and then we dissociate the cells and replate them again as a single cell suspension, and we do this for 3 passages. So we analyze the results at each passage and at the end of the 3 passages. So the first thing that we were able to observe, and the most important in this type of culture, is that there were no aggregate agglomeration. So this occurs most times when the culture media isn't robust enough to be able to keep the aggregates separated. So we want to ensure that we have a population of aggregates as homogeneous as possible. And in this case, we were able to have them. So we took several photos. We also used 2 different cell lines. So we used the Gipco episomal IPS cells, and we used the TC Lab, which is our in-house IPS cell line. So we analyzed the photos that we took at each passage, and we studied the behavior of the aggregate diameter. So as we can see, we have narrow populations of aggregates, either between the Gipco epi and the TC Lab, and we can see that we have an increase in the aggregate diameter at the last passage, SP3, but that's mainly because that's the only passage that we let cells grow for 5 days instead of 4. So nevertheless, it's very important that we didn't have any aggregate agglomeration. But besides the homogeneity of the population, we also wanted to assess if the cells were growing. So for that, we counted the cells at each passage, and we can see that there's an increase in fold change at each passage that goes up to 8.2-fold in only 5 days. So this would lead, after 3 passages, which were 14 days, to a cumulative fold change up to 140-fold, which is pretty good. But we did not want only the cells to be there, we want them to be healthy. So we tested if the cells remained viable, and we were able to have high percentages of cell viability for both cell lines for every passage that we tested. Also, we wanted to observe if the cells were still pluripotent. So one analysis that we performed was to replate the cells at the end of passage 3, and assess the pluripotency via immunocytochemistry by staining them for the pluripotency marker OCT4. And as you can see, almost all of the cells, if not all of the cells, are still positive for OCT4. So even then, one important thing when we are trying some long-term culture of pluripotent stem cells especially is to verify if the cells are stable. So for that, we use the karyostat assay, which allows us to assess the genomic stability of pluripotent stem cell lines. And so we tested samples after 3 passages for both cell lines, and as you can see, there's no chromosomal aberrations found, so the cells remain stable. So we verified that we could use stem scale as a great culture media to expand the cells in the orbital shaker. And next, we wanted to verify if we were able to use this for a scalable platform such as the PBS bioreactor. So for that, we use the PBS 0.1 MAC, which has a working volume of 100 ml, and we use the same seeding density and the same protocols. The only difference here is that for each passage, we established a limit of 5 days for every passage between dissociating the cells and seeding them again. So at the first glance, we can see that the population appears to be even more homogeneous than the ones that we obtained in the orbital shaker, and we were able to verify that by doing the aggregate diameter distribution. So we can see that the distribution is very narrow, and it's pretty similar between each passage and between each cell line. So, and we also observed that the average diameter did not surpass the 400 limits, which was our goal, and that the diameters that we obtained were pretty similar for both conditions and for each passage. And again, importantly, we did not observe any aggregate agglomeration. Again, we also wanted to assess if the cells were able to grow efficiently. And in this case, in the PBS bioreactor, we were able to obtain a fold change up to 9.3-fold in only 5 days, which would lead, at the end of 3 passages, or in this case 15 days, to a cumulative fold change that goes up to 664-fold. Also, when checking cell viability, there was high cell viability with more than 92% of viable cells in every passage for both cell lines. And in this case, we also wanted to assess, as we are working with aggregates, if the aggregates were indeed viable. So what we did was a live death assay where we could see that the majority of cells within the aggregates are live, and the cell death that we observe is not significant. So, and at the end of 3 passages, we also assessed if the cells were still pluripotent, first via flow cytometry by staining for OCT4, and there was still a high percentage of OCT4 positive cells after the 3 passages, which was further confirmed by replating the cells and staining them for OCT4 and analyzing them via immunocytochemistry. So after the 3 passages, the replated cells still express OCT4. And again, as we do in long-term culture of cell, we also analyze the karyotype, and there, like in the orbital shaker, there was also no chromosomal aberrations found. Just to finish up, we also wanted to compare our samples from the orbital shaker and the PBS bioreactor with HPSCs control to verify, to confirm that the cells were still pluripotent. So we used the Pluritest, which compares the transcriptional profile of a sample to a reference set that has more than 450 cell types and tissues. So as you can see, for both cell lines and for both orbital shaker and PBS bioreactor, the cells passed the Pluritest, which means that they are still pluripotent. So by gathering all this data, we confirm that the stem scale is not only suitable for suspension culture of human pluripotent stem cells, but it's also one of the greatest options that we currently have, even for scalable conditions such as spinner flasks, for example, or in this case, the PBS bioreactor, since it's capable of achieving high cell yields while maintaining the cells in a pluripotent state and with a stable karyotype. So this was the first part of our work, and next we wanted to verify not if the cells were able to expand as pluripotent stem cells, but if we could create a scalable process that will allow us to induce the pluripotent stem cells into the neural lineage. So to do that, we first start with an adherent pluripotent stem cell culture. We incubate them with Accutase to obtain a suspension of single cells, and then we generate the pluripotent stem cell aggregates. So, and from then, we go to the neural precursor cells. But there's only one small thing about this, is that the initial diameter of the pluripotent stem cells will be crucial towards the efficiency of derivation, especially for the neural induction. So we have previously determined that the optimal conditions to induce cells towards the neural lineage was to use pluripotent stem cell aggregates with an average diameter of 150 micrometers. So with those aggregates, and in only 7 days, we would be able to generate pure populations of neural precursor cells. So we wanted to do this not only at a culture plate level, but at a spinner flask level. So for that, we used the Selster-Witten 100 ml spinner flasks with a working volume of 80 ml. And to induce the cells towards the neural lineage, we used the Gibco pluripotent stem cell neural induction medium. So this is a serum-free culture medium that provides high efficiency neural induction. So it's a rapid induction, it's scalable, and allows us to generate high quality neural precursor cells. So this was designed for 2D culture, but we were able to adapt it to 3D culture in the spinner flasks. So I have here some of the photos that we took during the neural induction, and the first thing that we can observe is that by day 0 we have the pluripotent aggregates, which are round and have a homogeneous limit, but at day 1 we do have some structures arising. So the aggregate starts to bleb, and then smaller aggregates are released from the larger ones. And by day 6 and 7, we almost can see any of the original aggregates, but instead we have smaller structures, smaller aggregates spread all over the spinner flask. And one curious thing about this medium is that when we use, for example, the MB27, which is the culture medium that we classically used for the dual SMAD inhibition protocol, at day 6 of induction, we still have the round-shaped aggregates with a denser center. But what we see after the 6-7 days of induction using the NIM, the neural induction medium, is that we do have some differentiation structures that resemble a picobasally polarized tissue characteristic of the neural cells. And another great thing of using this medium, the NIM, is that using the B27, we usually obtain, after 6 days, a 3.5-fold change in the cell number. So with the NIM, we were able to increase this by 10-fold. So we were able to obtain around 35-fold change after 7 days of induction, which would lead us to have more than 300 million neuroprogenitors in only 180 ml spinner flasks. So, okay, we do obtain large numbers of cells, but how are the aggregates? So we also did a live and dead assay, And as you can see, most of the cells are alive, and we almost have no cell death within the aggregates, which is pretty good. So also, we further maturate the cells, and at day 34 of maturation, we are already able to observe TUJ1 positive neurons. Also, at day 34 of differentiation, we are also analyzed the calcium transients in order to evaluate the response to potassium chloride and histamine to allow us to functionally identify the neuroprogenitors, the neurons, astrocytes, and even immature cells. So from here, we can see that our population has around 62% of responding cells, and from that 62%, we have 20% of the cells that are already neurons, and 80% that are still on the progenitor phase. So this neural induction medium allows us to have large numbers of neural precursor cells that are able to differentiate into fully functional neurons. But still, we wanted to check if we could be able not only to derive the pluripotent stem cells into neural precursors in general, but if we could direct them into a specific neural lineage. So for that, we use the Pluripotent Stem Cell Dopaminergic Neuron Differentiation Kit. So this kit was especially designed to differentiate the pluripotent stem cells to functional midbrain dopaminergic neurons. So again, this culture medium was designed for 2D culture, and we were able to adapt it to 3D conditions. So we adapted it, and the first 6 plus 5 days, which are the floor plate specification and expansion, we did it on the cell-stir-Witten that we used, the same one that we use for the neural induction medium. But in here, we had to consider that for this specific lineage, the initial aggregate size would have to be around 200 micrometers. So we initiated the differentiation with that diameter. And a curious thing is that during the induction for floor plate progenitors, we can see that we also experience some membrane blebbing, like in the first days, but by day 6 and 7, we also have the structures that we observed with the NIM, but instead of the cells starting to divide into smaller aggregates, they remain in the original aggregates as more denser centers. So after the floor plate specification, we replated the cells and we stained them for the FOXA2 floor plate marker. And as you can see, almost all of the cells are expressing FOXA2. We also analyzed the aggregates that we obtained by day 11 doing a live dead assay, and again we have negligible cell death within the aggregates. And the curious thing is that we compared by real-time PCR samples from pluripotent stem cells, from cells recovered at day 11 using the dopaminergic neuron differentiation kit, and cells from day 7 using the neural induction medium. And as you can see in here, we have upregulation of PAX2 and EN2, which are midbrain markers, when we use the dopaminergic neuron kit, and when we use the neural induction medium, we have an upregulation of FOX2 and PAX6. So this proves that this culture media can be scalable and still generate midbrain progenitors using 3D suspension culture instead of using the regular 2D approach. So just to do a quick sum up of what we have been talking about, so StemScale is a promising new culture medium specially designed for pluripotent stem cell scalable suspension culture. So we can yield up to 664-fold in only 15 days. So at the same time, this culture medium StemScale can also maintain high levels of pluripotency markers and at the same time, maintain a normal karyotype. On the other hand, the neural induction medium also allows us to have up to a 32-fold increase in cell number, and the cells that we obtain are able to further maturate and stain positive for TUJ1 and also respond to potassium chloride and histamine treatments. Also, the pluripotent stem cell dopaminergic neuron differentiation kit was able to be used in a scalable platform using 3D cultures, and we were able to obtain pure populations of midbrain progenitors. So we are finishing here. I just want to acknowledge first Professor Tiago Fernandes and Professor Joaquim Cabral from our lab at IBB in Lisbon. And I would like to do a special thanks to all of the team of Thermo Fisher Scientific, especially to Michael Eckenhead and Michael Durr, who have been working with us for the past few years. So thank you all so much, and please continue to enjoy the 5 days of stem cells.